Welcome to Easy Microbiology.
Good evening, everyone. Today I am here to discuss our research into the bacteria Bordetella pertussis and its ability to replicate and survive outside the human body. We will be exploring its habitat, morphology, cultural characteristics, biochemical characteristics, and host range. I aim to provide you with a better understanding of the astonishing abilities of this bacteria. Let's begin. Bordetella pertussis is a unique and highly contagious bacterium that is responsible for causing whooping cough. It is transmitted mainly through respiratory droplets when an infected person coughs or sneezes. This bacterium is adapted to survive and replicate only within the human body, and is unable to survive outside of it. Gaining insight into its environment, characteristics, and host range is a crucial step in finding ways to fight against it. Bordetella pertussis is an obligate human pathogen as it only resides in the human respiratory tract and is unable to replicate nor survive outside the human body. Let us explore the morphology of this bacterium in the next slide. Bordetella pertussis is a gram-negative bacterium characterized by its small size and lack of motility. It has a cocobacillary shape, meaning it is round and rod-shaped, and is encapsulated by a capsule composed of complex sugars. This capsule is believed to contribute to the bacterium's survival both inside and outside of the human body. B. Pertussis is a fastidious organism, which needs specific growing circumstances in order to replicate. It can only reproduce on special media that includes blood and certain antibiotics in order to stop other types of bacteria. This restriction forms an atmosphere where B. pertussis can only persist and duplicate inside the human body, making it hard to survive and replicate in any other environment. Bordetella pertussis has some distinct biochemical features that make it unique. It is catalase positive, meaning it produces the enzyme catalase, and does not ferment carbohydrates. This property allows it to survive and propagate outside the human body. Bordetella pertussis specializes to humans and does not infect animals. It primarily infects infants and young children in the respiratory tract, which makes it difficult for the bacteria to replicate and survive outside the human body. Now we'll look at the next slide. B. Pertussis is a gram-negative bacteria that is specialized for survival in the human respiratory system. It produces various virulence factors such as pertussis toxin, filamentous hemagglutinin, adenylate cyclase toxin, tracheal cytotoxin, and dermonecrotic toxin, which allow the bacteria to colonize and cause damage in the human respiratory tract. Although these virulence factors enable B. pertussis to colonize the human respiratory system, it is difficult for the bacteria to replicate and survive outside of the human body. Bordetella pertussis is particularly resistant to antibiotics due to its limited antibiotic resistance mechanisms. In this case, the most common mechanism of resistance is through genetic mutations in target genes. One such gene, macrolide resistance protein A, has been identified as being especially important in providing the bacteria resistance to antibiotics. In addition, the bacteria has also been found to be more resistant when in the presence of certain environmental factors. This emphasizes the need to understand both the genetic and environmental elements that may contribute to the bacteria's ability to resist antibiotics and survive outside the human body. Epidemiology is the study of the distribution and determinants of health-related states or events in certain populations. With regards to Bordetella pertussis, a global emergence of the disease has been observed in recent years. In particular, the disease is observed to primarily affect infants and young children, and severe cases of infection can be life-threatening, especially in infants under 1 year of age. Vaccination is an effective tool to reduce the spread of this disease worldwide. B. Pertussis has the potential to colonize and cause damage to cells in the respiratory tract. It attaches to cilia in the airway epithelium and produces toxins which damage the ciliated cells, resulting in inflammation and mucus production, and thus causing the characteristic symptoms of pertussis, such as severe coughing fits. Furthermore, the toxins produced by B. pertussis also hinder the body's immune response, thus allowing the bacteria to be able to persist in the respiratory tract for longer and thus causing a longer duration of the infection. Our bodies depend on immunological responses to protect themselves from pertussis infection. Both adaptive and innate immunity, including IgA antibodies, help keep this bacterium from attaching and neutralizing toxins. Therefore, immunological responses can be considered a way to prevent the spread of this harmful bacteria. Collecting specimens from the respiratory tract, typically a nasopharyngeal swab or aspirate, is needed for laboratory diagnosis of Bordetella pertussis. These specimens are then tested using techniques such as PCR or culture, in order to detect the presence of the bacterium. Accurate diagnosis is important for proper treatment and prevention of further spread of the infection. PCR testing is preferred as it is a rapid, sensitive, and cost-effective method for diagnosing pertussis. Vaccines are highly effective at protecting against pertussis infection and mitigating its potential for spreading. DTaP is one of the most reliable and effective vaccines used to combat Bordetella pertussis. The vaccine should be administered at 5-dose intervals between 2 and 6 years old, with an additional shot administered around 11 years of age. Vaccinating both children and adults is the most effective way to protect against this potentially serious bacterial infection. Antibiotics like macrolides and trimethoprim sulfamethoxazole have been revealed as possible treatments for Bordetella pertussis. Early start of treatment is established to reduce the intensity and length of signs and symptoms tremendously. Studies show that treatment is most reliable when carried out in the initial phase of illness, before the bacteria has the capability to spread. Moreover, using antibiotics can reduce the amount of potentially contagious people, aiding to prevent additional transmission of the disease. Bordetella pertussis is a highly contagious bacterium that can cause serious respiratory illnesses. To effectively manage and control the disease, it is important to understand its habitat, morphology, cultural characteristics, biochemical characteristics, and virulence factors. Furthermore, studying the epidemiology, pathogenesis, immunological responses, diagnosis, prevention, and treatment of this bacterium is critical to better protect public health.
Thank you for watching. If you enjoyed this video, please like and subscribe for more content.